The Development of Embryonic Stem Cells by Kelly Watkins. Scientists have learned how to grow and divide embryonic stem cells in a culture dish to produce stages that will eventually provide researchers with immaculate results. Stem Cells 101 explains that stem cells are unspecialized cells that can renew themselves through cell division for long periods of time and can mature into muscle cells, nerve cells, and bone cells. Embryonic stem cells are multiple cells derived from a 5-day pre-implantation embryo that develops into cells and tissues. Embryonic stem cells come from a blastocyte, or a small sphere of cells from cell division in a fertilized egg. Scientists harvest these cells from the inner cell mass of the blastocyte when the cell is 6 days old and consists of 200 cells. Growing cells in the laboratory is known as a cell culture. According to Watkins, for the purposes of stem cell research, human embryonic stem cells are cultured in the laboratory by moving the inner cell mass into an animal cell culture dish that is rich in nutrients and growth factors. The animal cell culture dish, or also known as feeder cells, are a coated layer of mouse embryonic skin cells, which helps the embryonic stem cells to attach and grow. Embryonic stem cells can divide continuously over the surface of the culture dish. Many days later, embryonic stem cells multiply and begin to crowd the culture dish. During this stage, human embryonic stem cells are gently removed and plated into many different animal cell culture dishes. This process is called subculturing and is done many times for several months, since these delicate little cells tend to get crowded in the culture dish. Every time scientists subculture the embryonic stem cells, each cycle is described as a passage. After approximately 6 months, the original 30 cells of the embryonic stem cells produce millions of stem cells. Once these genetically stable embryonic stem cells are established, these batches of cells can be frozen and shipped to other laboratories for further culturing for stem cell purposes, or continuing their journey in the culture dish subculturing. If these cells are allowed to clump together, they will form embryonic bodies. Once these types of cells continue to grow, these embryonic bodies can form muscle cells, nerve cells, and other cell types. Scientists try to control these generated cultures by changing the chemical composition of the culture, altering the culture dish, or modifying these cells by inserting specific genes. Scientists can direct these cells into specific cell types, which may result into treating certain diseases. If these stem cells can be made to go to suitable places in the body, and they may become the suitable cell type. Stem cells could be made to migrate into damaged heart muscles and could become heart muscle cells and eventually cure the damaged heart muscles. This is only a short overview about the development of embryonic stem cells. In the laboratory, it would take professional scientists many months to configure these little supercells. However, researchers have come a long way to providing treatment for our future in medicine. For more information, please visit www.stemcellresearch.org or www.embryonicStemCellResearch.org. Thank you.